Hi everyone, this is Team 15 and welcome to our final presentation of the blood cell classifier. The goal of our project is to distinguish and classify different blood cells in a blood test sample to assist in clinical diagnosis. Traditional blood-based diagnosis usually includes identifying and counting different types of blood cells, where the laboratory workers need to manually perform these tests using different apparatus and chemicals, which has been time-consuming, tiresome, and dependent on the worker's experience. Machine learning algorithms can be applied to automatically identify and classify the cell subtypes in the blood sample, with the existence of a large and reliable dataset that can be used to train our model. These are both motivations for our project to create an effective and reliable machine learning model, replacing human labor in identifying and categorizing various cell types in the blood sample with different nucleus structures into four groups: monocytes, eosinophil, lymphocyte, and neutrophil, thus dramatically increasing the efficiency and accuracy of clinical diagnosis.
We initially download around 12,500 blood cell images from the Kaggle. As you can see in the background, most of the images clearly show the structural features of the cells, and each image contains one type of white blood cell. Therefore, we can use the shape difference of the nucleus to train our model and do the classification. First, we resize the image to the pixel size of 224 by 224 to better train our model. Later on, we noticed that the bright blood cells in the background influence the model during the training stage. Because the identification of white blood cell is mainly used on the structural features of the nucleus. Therefore, after extensive research, we found out that the contrast stretching can enhance the blood cell images. Contrast stretching is an image enhancement technique. It is stretching the range of pixels to a larger range so that the shape of the nucleus is enhanced and the outline of the red blood cell is weakened. However, there are some corrupted images in the dataset. Some images are incomplete and some images fail in the contrast stretching. As a result, the structure features of the nucleus completely disappear. We eliminate most of the corrupted images, but it is hard to guarantee a perfect dataset due to the large amount of data. Later on, we split the dataset into training, validation, and testing data to train and evaluate our model.
Our team utilized a support vector machine classifier as the baseline model. We trained two most common kernels, which are RBF and polynomials, using the same dataset. Eventually, we got the highest test accuracy as 24.5%. For our primary model, the team has extensively trained lots of different hyperparameters, and we document their impacts on the training and validation accuracy. Particularly, the team tried different feature extractors such as BGG16 and AlexNet. We also used dropout and weight decay in our model to cope with the overfitting problem. Eventually, our team decided to use pre-trained AlexNet feature extractors to acquire the feature maps of our data and then train our own classifier that is made of one CNN and two fully connected layers. Here's the diagram of our architecture. The process image is fed into the AlexNet, which then outputted pre-trained features and delivered to our own neural network for final prediction.
The training curve shows that our final model can achieve 99.8% training accuracy and 19.7% validation accuracy. The loss curve shows that the training loss almost reached zero after 100 epochs, while validation accuracy stabilized at 19.7%. Test accuracy is found to be 61.2%, partially due to the overfitting problem. The confusion matrices are used to evaluate our model's abilities in recognizing different blood cells. The confusion matrix for the test dataset shows that the lymphocyte and neutrophil have the best test performance, whereas eosinophil and monocyte have the poorest performance in that they both have a large number of false negative cases of neutrophils. Lymphocyte and neutrophil are the two most accurately identified cell types because they have distinct and unique features that can be accurately captured by our model. For example, lymphocytes are round and neutrophils have two lobe nuclei. Some cell images have lost their unique features after processing, which create confusing cases that result in inaccurate predictions. Moreover, blood cells of the same type can have very different colors of their nuclei. Therefore, the team has found that the randomness in color intensity greatly reduces our model's accuracy.
Now, we'll show a demonstration of our model. The lab worker will upload a microscopic blood cells image onto the folder as shown here. In our demo, we will put 12 random images from the test sets which is never seen before by our model. Then let's open our program. By changing the directory of our image folder, we can start running our program. The program will automatically process the image and feed into AlexNet and eventually be predicted by our neural network. For demonstration purpose, we'll print out the images and its corresponding true and predicted label for comparison. The result shows an overall accuracy of 66.7%. As you may have seen, a CSV file has been simultaneously generated. This is a cells report that contains the total number of each type of white blood cells, which serves our ultimate goal of increasing the efficiency of blood cells counting.
Overall, this project was a great learning experience as we implement machine learning into real-world applications with decent performance. Reflecting on the project, we encountered several unexpected challenges at the beginning. For example, it was difficult to emphasize the nuclear structure of the white blood cell in the raw images. In addition, flaws in the image quality include blurring and loss of blood cell features due to contrast stretching, negatively impacted the model accuracy. Model selection and training also posed challenges as we found it hard to further increase the test accuracy to beyond 90%. With future iterations addressing these aspects, we believe that this application of machine learning on blood cell classification will benefit the clinical diagnosis and make a positive impact on society.